Okay, good afternoon, everybody. Welcome to Virology. Does anybody know what virus is up here on the screen? Exactly. But that's a good guess, though. What else would I put up as the first slide of the course? That's swine influenza. It happens to be, in fact, the strain that has caused the pandemic in the past year. And we're going to be talking a lot about this. You're going to know by the end of this course exactly where this virus came from, how it arose, why it causes disease, what's inside of it. You're going to know that it has negative stranded RNA in it. You're going to know all about its detailed structure, and all because this is important for you understanding how this virus causes disease. And there are many, many other viruses that we're going to talk about today. There are thousands upon thousands of viruses out there, and these are just some of them in this cartoon. And by the end of this course, you should be able to look at some of these and go, that is a baculovirus up there, that's a pox virus, that's an adenovirus, and understand how they cause disease. My goal with this course is to have you go out into the world and understand viruses and what they are doing in the world, how they make you sick and how some of them don't make you sick. Because very few people understand viruses at any level, as was exemplified by the events of the past year. Every journalist just about got swine flu wrong in all the articles they wrote. And I want you to be able to go out there and read an article in the Times in 5 years and say, that's not right, and know why. So that's one of the goals of this course. So this is Biology 3310, Virology. Anybody here ever had a virology course? No. There hasn't been a virology course taught here since the mid-'80s. I know because I taught in it. And it stopped for some reason. I don't actually have my lab on this campus. We're at the medical center, so we were guests in that course. And I think it's a shame that there hasn't been a virology course here for so many years. So this is my effort to try and fix that. So let me tell you a little bit of organizational information about the course. I'm Vincent Racaniello, and that's my email where you can always reach me. I work at the medical center. My lab is up there. I'll be coming down here to teach this course twice a week. I'll be assisted. I'm also on Twitter, by the way, which I use extensively to look for and disseminate information about viruses. I found it to be a really powerful tool. I follow only scientists, and it's where I get a lot of my information. And we might talk about a particular story in class that I've picked up on Twitter. So if you want to see what I'm talking about, it's all science. You might want to follow me. I'm going to be helped in this course by Professor Saul Silverstein, who's sitting up. Can you stand up, Professor, and let everyone see how tall you are? There you go. So Professor Silverstein is my colleague at Columbia, up at the medical center. Between the two of us, we have 60 years or more experience working with viruses. So that alone wouldn't make us qualified to teach virology, because not all good scientists are good teachers. But we happen to like teaching as well, and that's why we're doing this course. And Saul's email is there as well. And we will be assisted by a TA, Esther Francisco. Please stand up, Esther. Esther's a PhD student in my laboratory, a former undergraduate on this campus as well. So she'll be helping us out, and her email here as well. So throughout the course, if you have any questions, don't hesitate to ask any of us. We'll see if we can answer your questions, because the thing about viruses is that we don't always have an answer to all the questions. Now, I'm going to be posting a lot of material for the course, either at Courseworks or on the course website, which I maintain myself, so they're both going to be the same. Some people I know like one. I don't particularly like Courseworks, so I keep a website. But you can go to either one. And there will be, of course, the schedule, what the topics are going to be for each lecture, what you should be reading for each lecture, and also screencasts. I'm going to be recording all of these in sync with the slides, and I'll be putting them up every evening after the lecture, again, either at Courseworks or at the website. So if you fall asleep, you can always hear me later on. And they'll be downloadable. You can put them on your iPod or whatever your media player of choice might be. So those are some of the resources. The textbook is Principles of Virology, of which I was a co-author. So I think it's a very good book, obviously. It arranges virology according to the steps in the infectious cycle. It's not an encyclopedia. It doesn't have a chapter on influenza and a chapter on herpes, because I don't think that's the best way to first learn virology. I think by process, attachment to cells, entry, uncoding, nucleic acid synthesis, I think that's how you really learn the fundamentals, and then you can go on and learn about specific viruses. So in this course, we're going to be discussing the individual steps in the infectious cycle. And there are specific chapters in the book that will correspond to each session, and I'll post those again on the web so that you can read it. There are also a couple of other resources I would really like you to read, and one of them is a blog which I maintain. It's called Virology Blog, and I do about 2 posts a week. This is really aimed at a non-science audience, so I keep it short, 500 words or less per post, and I explain viruses. So if something happens in the news, I write a post about it. If I see a paper that's interesting, I write a post about it. These will supplement what I tell you here. So both the textbook and the blog will supplement the information I put here. It will extend using new examples, for example, or it will make something clearer. And my philosophy about learning virology is the more you immerse yourself in it, the better you'll learn it. You don't necessarily have to memorize anything, but I feel if you read and listen to virology, you'll learn it better. And in terms of listening, I also do a podcast called This Week in Virology, and this is a weekly show which is on iTunes, and it's on the web as well. And I sit down with a couple of virologists and we talk. You can imagine we're in my home sitting on our couch. You probably know Dignation, most of you. These guys sitting on a couch talking about tech. Well, we sit on chairs in my office and talk about viruses. We record it and we put it up. We make it accessible. I have guests joining the conversation via Skype, and I think it's very interesting, and I would highly recommend that you listen to it. Again, it's on iTunes. It's always among the top 10 in the science and medicine category on iTunes. We have thousands of listeners, and so I highly recommend that you take a look at it as well. This is an example of a blog post that I did recently. This is called Bornavirus DNA in the Mammalian Genome. So a paper came out just a few weeks ago showing that the chromosomal DNA of several mammals, including humans, were found to have sequences related to the nucleoprotein gene of a Bornavirus, which are envelope viruses with negative strand RNA genomes integrated in the chromosome. And I wrote, I am amazed by this finding, because I am. How did the DNA get there, and what is it doing there? So the post is pretty short, digestible. And so this is the kind of information that will complement what I'm talking. Now, one of the best parts about all this online material is that it's interactive. I have comment sections at the bottom. So if you go down at the bottom of these posts, you will see that people from everywhere post comments. And some of them ask questions. Some of them are anti-science types who say we don't know what we're talking about. But for the most part, they're really good questions, and they can have a lot of discussion. And when I deal with or write about viruses of medical importance, you should see the comments from people who have these illnesses and want to know what they can do. So for example, chronic fatigue syndrome has recently been associated with a specific virus, and I've done a few posts on those, and the patients come and they post and they ask questions. So it's very interesting. It's real-world virology. So that's the blog. And then there's the podcast, which also has a website, This Week in Virology. And last week, we did an episode with Matt Freeman, who is a new assistant professor at the University of Maryland, and he's doing this great project where he goes out into Western Maryland, he traps bats. He gets feces from the bats, he brings it back to his lab, and he sequences it to look for viruses. Now, this is something we never did 20 or 30 years ago because sequencing was too expensive and too tedious, but now you can sequence millions of bases in a few days. And so you can take total RNA from almost any sample, sequence it, throw away the ribosomal RNA, all the cellular genes, and see if there's any virus in there. So he's doing that because bats are a very big reservoir of viruses. And in fact, that's where the SARS coronavirus originated, from a bat in China. So that's the kind of conversation we have. We ask him questions, we talk back and forth, and again, we get a lot of interactivity with the world. People send in questions and we read them on the air. So I think you'll enjoy it. Again, you can go to iTunes and download it. And if I have particular episodes that are relevant to a lecture, I'll recommend them to you. So you have a lot of resources to build upon. You have, oh, one more thing which I didn't tell you. Before every lecture, I'll post the slides on the websites, and you can download them and print them and take notes if you'd like. You don't have to do that. It's there if you want. But then afterwards, I will post all the slides again with audio. So you have lots of resources, and you can go back to them. All the information I want you to be familiar with is going to be in front of you. The textbook, the lectures, and the notes, the blog, and the podcast. Now, what about viruses? Well, viruses are everywhere. This room is filled with them. In fact, all living things survive in a sea of viruses, as you will see, more than you ever imagined. We eat and breathe billions of them regularly. They're everywhere. We breathe 6 liters of air a minute, which is a lot, and we eat thousands of grams of food and its contaminants. We touch all kinds of things. The doorknob is probably covered with viruses. And we're pretty much okay most of the time. But we've got lots of viruses and lots of ways to introduce them to us. Even an innocuous place like the ocean is full of viruses. There are over 1 million virus particles per milliliter of seawater. And we'll talk about what that means and how that was discovered. We carry viral genomes as part of our genetic material. I just showed you an article about bornavirus DNA in our genome. That's very unusual because up until that paper was published, the only DNA of viruses in our genomes that we knew about was retroviral DNA. We have thousands and thousands of copies of retroviral genomes integrated into our chromosomes and also that of many other animals as well. Viruses infect our pets, our domestic food animals, wildlife, plants, everything. Anything living has a virus that infects it. And virus infections of animals can cross over into humans and vice versa. They go back and forth. And that is called a zoonotic infection, a virus infection of an animal that infects a human. So influenza is a zoonotic infection. When new influenza viruses arise, they come from animals. And so it's a zoonoses. So viruses are everywhere. The numbers are staggering, and this is something that I will keep emphasizing over and over. And one of the reasons that viruses are so successful is because they are present in huge numbers. So let's take one example. This is a bacterial virus, which we call bacteriophage. It's a virus that infects bacteria, carries its nucleic acid in this wonderful head structure. And here it's shown on the surface of a bacterium injecting the DNA through its neck and tail into the bacterium. It attaches to the bacteria by these wonderful looking legs here. It turns out that there are over 10 to the 30th bacteriophage particles in the waters of the world. 10 to the 30th. That's a huge number. How huge is it? Well, a particle of a bacteriophage on the average weighs about a femtogram, 10 to the minus 15 grams. So we can do a little math here and calculate the weight of all the bacteriophages in the world. And remember, these are things you can't see. So if you do the calculation, you find that the biomass on Earth of bacterial viruses alone, not other viruses, exceeds that of elephants by more than 1,000-fold. Now, an elephant is something you can imagine, right? It's big, weighs thousands of pounds. And there's more phages on the Earth by weight than elephants. That is amazing. So they have to have some role or else they wouldn't be here. And we're going to learn about that. Now, what if you line them all up in a single line? How far would they reach? 200 million light years. This is an astounding number. A light year, right, is how far you can travel for a year going at the speed of light, which is 186,000 miles per second, right? 200 million years is how long this line of phages would stretch. And if you don't believe that, you can go to this website. The fellow named Phage Hunter has made the calculation, and that's where I got the numbers from. How far do those phages stretch? It actually was a calculation suggested a while ago by a virologist, and he actually redid the numbers so you can check it out for yourself. So what's 200 million light years? I have no sense for. Well, the nearest galaxy to us is Andromeda, and that's 2.5 million light years away. And you can't even see it with your eye in the sky. So we're in the Milky Way. This is the nearest galaxy. So it's 100 times farther, the phage line would be stretching into space. So the point is that there are a lot of phages in the waters of the world. Now, whales, here's another great example of the source of viruses. Whales are commonly infected by a virus which is shown here. It's called a calicivirus. It's relatively small as viruses go. We'll talk all about the size. Very common infections of whales. When people go out in the oceans and sample whales, they find caliciviruses. These viruses cause a variety of ailments in whales: rashes, blisters, gastroenteritis, also in other mammals of the sea, dolphins, seals, and so forth. And the numbers are astounding. The viruses can infect us, and that's been known to occur. The whales secrete over 10 to the 13th caliciviruses a day. 10 to the 13th, not 10 to the 30th. It's not that big, but this is a big number. So in virology, we always deal with exponential numbers because the numbers would be too long to do otherwise. So one whale excretes this much virus, and some of those viruses may infect people. So you can see there are many opportunities for viruses to cross species. Here's another example. Yes?
How will the whales secrete the virus? Just through, spouting?
No, this is a gastroenteritis virus. It replicates in the intestines, so it would be shed in the whale feces. Okay. And then other whales would pick it up because they filter water, and other mammals, other animals, and even humans. You know, you go for a swim at the ocean and you take water in your mouth, right? You spit it at your friend. There's a lot of viruses in there. So, you know, I learned about this 5 years ago, and I haven't done that since. So, but you know, we all grew up doing that. So I think the danger is minimal, unless you're immunocompromised, and that's another story. Here's another number: 10 to the 16th genomes of human immunodeficiency virus on the planet today as we speak. Another huge number. All the infected people have millions and millions of genomes in them. It adds up to 10 to the 16th. Now, what that number means, because these viruses all make a lot of mistakes as they replicate, as we will learn in this course, what that means is that there are on the planet today genomes that are resistant to all the drugs we have that we use against HIV, and there are over 20 of them. So resistance is already out there for all of those drugs, plus any other that we would ever develop. The number is so large that you can't ever make enough drugs to cover all the resistant mutants that are out there. So it's almost futile in a way. You can never win, but you can try and keep up. Amazingly, though, despite having all these viruses, and there are many more, these are just some examples to give you a sense, we're pretty healthy most of the time. You know, this room is full of viruses. I'm sure if we sampled it, it would have viruses in it. I always wanted to sample the subway bars and the cars, but I think if I went in with a swab and a tube, I would eventually be told to stop. But I bet there are tons of viruses on there. And that's because we have a great immune system. We have a defense system which helps us fight infections. How many people here took Moshewitz's immunology course? Great. So you have a really good foundation in immunology, and we're going to understand how that relates specifically to virus infections. Now, when your defense system is down, when you have AIDS, you become immunocompromised because HIV replicates in cells of the immune system. Or if you're immunosuppressed, if you're receiving an organ transplant, you receive drugs that suppress your immune response so you don't reject the organ. Unfortunately, you can't defend yourself against pathogens. And under those conditions, common viral infections can be lethal. And some of us have viruses in us all the time, and we're perfectly healthy, but if we become immunosuppressed, we develop disease. And we'll talk about some examples of that. So our immune system is absolutely essential for keeping us healthy in the face of all of these viruses. How infected are we? I'll give you lots of examples in this course, but let's start with one, herpes viruses. There are a number of human herpes viruses, which happen to be the viruses that my colleague Professor Silverstein has worked on his entire career. And here's an example of one. Every one of you in this room, I would bet, is probably infected with at least two of the known human herpes viruses. And here they are: herpes simplex 1 and 2, varicella zoster virus, cytomegalovirus, Epstein-Barr virus, and human herpes viruses 6, 7, and 8. There's another human herpes virus, B virus, but since it's 100% lethal, I doubt that any of you have it in you. So I think just by looking at the results of serological surveys of populations, it's likely that most people have at least 2 of these viruses. Now, a defining characteristic of the herpes viruses is that once you're infected, they're with you forever. You can't get rid of them. You become latently infected, and that's something you'll hear about more from Professor Silverstein. As I mentioned earlier, every one of your cells is also infected with viruses. We have these viral remnants in our genomes, and each of you has thousands of copies of old and new retrovirus genomes integrated into your DNA. It makes up about 8% of your DNA. And in fact, it is what distinguishes us in our nucleotide sequence of our chromosomes from chimpanzees. So we are about 95% identical to chimpanzees in our DNA sequence, and the 5% difference is mostly these retroviruses. And I once saw someone walking around the lab with a T-shirt that said 95% chimp, and that's correct, because we are. And it's all viruses that make up those differences. So why are they there? And why have they been there for millions and millions of years? We'll see if we can answer some of these questions. You will pass these genomes on to your children, and they'll do the same with theirs. They're in our germline, so we pass them on. What are they doing there? How did they get there? These are things that we're going to discuss. You're a reservoir for viruses that live in all kinds of places in your body. The lung is a very common place, the gastrointestinal tract. There are a few other locations in your body that harbor many viruses. We have adenoviruses in us. We're colonized. A lot of us are infected and shed them continuously. Coronaviruses, the family of which SARS was a member. Rhinoviruses, common cold viruses. Our guts are loaded with bacteria, which in turn have their own viruses. And you probably have heard that we have more bacteria in us than cells that make us up. And all of those bacteria have viruses of their own, either quiescent viruses that don't do anything or viruses that kill the bacteria. They've basically been with us since we've evolved, since the beginning of our existence. And they were clearly around long before, but that's a subject that we don't know very much about. So what I've been trying to emphasize to you is that viruses are amazing, and that's why we're teaching this course, because we think they're great. We want to impart some of that information to you. Yes?
I know they're like pro-bacteria, which are helpful for you, like you need them to survive, but there are also like pro-viruses that you need to have in you in order.
I'll bet there are. We don't know. It's a good question, because this is just a beginning area of study, and people are just doing surveys of humans for different kinds of bacteria. I would bet that they are beneficial. They confer some property to the bacteria that allow them to help you. Absolutely. And that's one theme that you'll hear in this course, and that viruses don't just make you sick, but they have roles in the world. They do good things for animals. They do good things for ecosystems. So yes, the answer is probably yes, although we don't know, because there hasn't been any work done yet. So the way we've designed this course is to help you see the big picture of virology. I want you to think about virology as an integrated discipline. I don't want you to memorize viruses, genes, diseases. I want you to put it all together, which is a little more difficult. I want you to see what goes on in cells infected by viruses everywhere throughout the ecosystem, the amazing machinations that viruses undertake in order to propagate. I want you to really appreciate that. And this is a really important point. I want you to learn the fundamentals about viruses that amaze the informed and frighten those who don't understand. So most of the world doesn't understand how viruses work, so they're frightened by them. And I want you to come out of this course with an understanding, and for the rest of your lives you will not be frightened by viruses. Yes?
Why do you call them molecular wizards?
Well, you'll see in the course. But they take over cells, essentially, in a myriad of different ways. And you will end up saying, how do they do all these different things and why? It's just amazing the different strategies that viruses have evolved to replicate. And that's why they're wizards, because they do many different things. Not because it's magic or anything, but because they do a number of, so many different strategies to replicate. So virology, to really understand virology requires you to know a lot about other fields in biology. And I assume that a lot of you have had biology courses here. I know a lot of you have had immunology, so that's good. Because virology always tests your ability to think and put diverse information together. You're not going to just think about the virus, but also what the cell is doing and how the virus interacts with the cell. You have to memorize some facts. You have to know that influenza virus, the picture I showed you at the beginning, is a negative stranded RNA. Because once you know that, you understand pages and pages about how the virus replicates. But you can't memorize all the different combinations of facts, and that's what really defines viruses. You'll have to think. You'll have to say, okay, this is influenza, it's got a negative stranded RNA, therefore when it gets into a cell it does this and this and this. And you'll be able to extrapolate by doing that. All the details are the most pleasing parts. That's the wizardry of viruses. All the strategies and tactics that they undertake when they infect a cell. Now, while we're talking about facts, I want to point out this nice quote by Clive Thompson, who writes often for Wired magazine. Sometimes they have good science in Wired, not always. But he says one of the reasons kids get bored by science is that too many teachers present it as a fusty collection of facts for memorization. This is precisely wrong. Science isn't about facts. It's about the quest for facts, the scientific method, the process by which we hash through confusing thickets of ignorance. It's dynamic, argumentative, collaborative, competitive, filled with flashes of crazy excitement and hours of drudge work, and driven by ego, or a desire to be the one who figures it out, at least for now. So I don't want to teach you facts. I want to teach you some facts, but I want you to put the pieces together. We will tell you how some of the facts were discovered by showing you experiments. We can't do it all the time because we don't have time in this course. But the key here is really ego, and I want you to leave this course with the understanding that science is often driven by the egos of scientists. Now, we all think we are helping humankind by curing diseases and making antivirals. But in fact, most scientists never get to do that. What drives them is getting a paper published in a prominent journal, getting a big grant, and doing better than their competition. And I think it's unfortunate that those properties drive science, but that's the truth. I've done it for over 30 years. It ain't pretty, but that's the fact. So when you factor that in, then you can understand a lot of the behavior of scientists. But it makes the system work to a certain extent. It also makes for ugliness, but I don't think that will change. Now, viruses, as I've said, infect everything, so they are a significant part of the ecosystem. And we've not appreciated this up until the last, say, 5 or 10 years, because many of the viruses out there we couldn't grow in culture systems. So we've had to sequence samples from out there and see the viruses are present. So, for example, Craig Venter went out into the ocean on his yacht. Craig Venter was the man who sequenced the human genome. And he got very wealthy as a consequence. He bought a yacht and he went sailing around the world and he collected seawater and sequenced it. And that's how we know that there are 1 million viruses per milliliter of water. But we couldn't do that 10 years ago. Now we do that. We know there are lots of viruses there. We're beginning to understand that they play big roles in our ecosystem. So all the viruses in the ocean, for example, have a big role in the turnover of carbon, the carbon cycle. They kill billions of bacteria a day, which liberates carbon for other organisms. So it's things like that that I want you to appreciate, not just that viruses cause disease. But if you don't know any of this, you think viruses are bad news wrapped in a bit of protein, right? The only news that makes it on TV or in papers or on the internet is bad news about viruses, killing people, making you sick. Very rarely do we see articles about all the good things that they do. And believe me, viruses do more than cause disease. And that's another thing I hope you get out of this course. So the big picture, again, which is what I want to teach you in this course, that all the diversity I'm telling you about, all these billions of viruses and different kinds and structures, we can actually look for common features and understand them that way, unity and diversity. So the basic thesis of this course is that all viruses have a very simple 3-part general strategy to ensure that they exist. I don't like using survival because that implies they're living, but you have to forgive me because it's the best word that I can come up with. 1, all viruses package their genomes inside a particle, which serves one function only, to get the genome from one host to the next. That's all the particle does. This is something that almost all viruses do. There are a few exceptions. but almost all viruses do that. Secondly, the genome contains all the information you need to complete an infectious cycle. That is a round of replication in the cell. We'll talk about that in great detail. We have an entire lecture devoted to it within a susceptible and permissive cell. And finally, all genomes can establish themselves in a host population so that the survival of the virus is ensured. So these three properties are shared by all viruses that we'll talk about. Okay, so if that's all there is to it, why do we have to study it? If it's just a matter of viruses transmitting and surviving, isn't there anything more? Well, there's a huge diversity of tactics, as I've said. That's what I referred to as wizardry before. And there are countless particles out there with amazing diversity. The size and nature and the structure of the genomes differ. We have very odd particles, as we'll see in this course. We have amazing coding strategies. So you're probably familiar with cellular messenger RNAs, where you have a stretch of sequence encoding for a protein. The protein is made, that's the end of it. Well, viruses break all the rules. They have amazing ways to coax the maximum number of proteins out of a messenger RNA. And they have incredible diversity in the cells and the organs in the animals that they infect. Finally, they can vary from not doing anything when they infect an animal, not causing any disease, being benign, or they can be lethal. So the virology is interesting because we have to explain all of these weird properties. How can viruses have a simple three-part strategy yet be so diverse in all of these steps? Now, there still is a simplicity underlying all of this complexity, which I will go back to over and over again in this course, so that you can understand viruses better. And that is two things. First, all viral genomes are obligate molecular parasites, and they can only function once they get in a cell. So no virus particle or a viral genome does anything until it gets into a cell. It has to do that in order to reproduce. And so I call them molecular parasites. You could call them cellular parasites. Either one works. And the second one, which is possibly even more useful, is that all viruses make messenger RNA that can be translated by host ribosomes. They're all parasites of the host's protein synthetic machinery. No virus can translate protein on its own. It needs the cell to do that. It's a very complex process. We'll talk a bit about it in detail, and only cells carry it out. No virus does any part of the translation machinery. There are some viruses, interesting, that encode parts of the translation machinery, and we don't really know why that is. These are pretty recently discovered viruses, but we don't know if that has any consequence. So no virus can do the whole thing by itself. It's dependent upon the cell to make protein. So if you can remember that, every virus needs to make mRNA that the cell will recognize. You will go a long way in understanding how viral replication cycles work. And in the end, really all the wizardry is great, but the best part is that it reveals the way the virus interacts with the cell. Yes?
Are all viruses parasitic, or do some of them have symbiotic relations with their host cells?
Well, a parasite has a symbiotic relationship, and usually we think of a parasite as causing a detriment to the host, right? So the answer is no, because what I'm trying to tell you is that not all relationships are detrimental. Not all virus-host relationships are bad for the host. And I'm going to tell you about some of these today, not today in this course, amazing ones where the host benefits by having the virus around and vice versa. So our classical notion was, yes, that the virus hurt the host, but in recent years we've learned that that's not the case. So the big diversity of viral strategies always reveals something about the host. So the virus has to do something to take advantage of a host process. So as we learn what the viruses do in this course, you're going to learn how the host works even better than you ever did before. You're going to learn about how cells take molecules into cells, how translation occurs, how trafficking occurs in the cell, and all because we can probe it with virus infections. We have to understand that to understand viral replication cycles. So you're not just learning about virology here, you're learning about the cells as well. Now, a word of warning. Be careful about anthropomorphic characterizations of viruses. What do I mean by that? Viruses don't think. Well, you say obviously they don't have brains, but people often say a virus will employ this strategy or ensure its survival, exhibit, display, etc. There are many, many words. In fact, there's a little bit of text in chapter 1 that deals with all the words that we don't like to use. Because the point is, and it's not just an exercise, it gets you to thinking about viruses as passive vehicles. Viruses have evolved to undergo chemical reactions which propagate them. They don't want to do something. They don't want to kill a host. They don't want to make a lot of particles. It's just a chemical reaction. And I think if you avoid using human-like words to characterize what viruses do, you set yourself in a better frame of mind for understanding how viral replication cycles occur. It's very hard to do. I'll slip all the time in talking to you. And in fact, you can probably find in our textbook all the mistakes we made where we left viruses doing something in. But I really think it's a good exercise to undertake to get you thinking about how viruses work in the correct way so they don't achieve their goals in a human-centered manner. And here's the key. They survive because they make huge numbers of mutants at every replication cycle. Those big numbers that I showed you earlier, they're all a consequence of making a lot of particles every replication cycle, more than any other form of existence on Earth. They make more of themselves, more progeny in a given time than anything else. And that's why they're successful, because a lot of those particles are no good. They don't work under certain conditions. The ill-adapted are gone. They never replicate. And if a condition changes in an animal or a host species, there are viruses there that can take advantage of that because there are so many of them, because there are millions and millions of particles produced in every cycle. There's always one that can take advantage of a different condition. A different animal walking by, a different weather, ultraviolet rays, you name it. It's really a beautiful example of selection of the fittest, and they're great Darwinian machines. I think there's no better model for the concept of survival of the fittest. So if Darwin knew about viruses, that's what he would have studied, because it's a lot easier than cataloging birds on a remote island. He can grow billions of viruses in a day and watch evolution occur in your cell cultures. So let's think about this a little bit more. Viruses, of course, depend on their host for survival. If there weren't any hosts on Earth, or if there weren't life, there wouldn't be any viruses. So what does that mean? If the virus is too successful, if it kills their host, then it doesn't have a host to grow in, right? On the other hand, if viruses are too passive and the host defenses overcome an infection, for example, the virus can be eliminated as well. So it's a little paradoxical, right? You have to have a balance between killing and existing with the host. And this has been debated by many, many people for years. It's a common misconception that viruses evolved to get more virulent for humans. So I saw in the past year in almost every newspaper the notion that this influenza pandemic strain was going to evolve to get more virulent in people. Why would it do that? It makes no sense whatsoever. Why would it kill and debilitate the host and affect its spread? Now, I'm giving a little anthropomorphic characterization here. I don't mean to. But in terms of selection and propagation, it makes more sense for the virus to coexist peacefully with the host. There are examples of virus infections that are terribly lethal, but they, for the most part, don't spread very much. Yes?
Right now, do they think the bacteria was before the viruses, or where do they place them?
That depends who you ask. You'll get both answers. Some people think, well, first there was a soup of, before any life, there was an organic soup from which life evolved. Some people think viruses could actually replicate in that soup before there were cells. So that's one, because theoretically it's possible. You could have just a simple nucleic acid genome multiplying in an organic soup. Others think that cellular life came first. It's a big debate because we don't have fossils like we do for a lot of other evolutionary questions. But it's a lot of fun to talk about. We're going to talk about evolution, and we'll do that. So how do you balance this? Survival of the host and survival of the virus. This is a really important underlying question. You don't want to kill the hosts off because then you have no hosts to multiply in, but you can't be wimpy, otherwise you're not going to get anywhere as well. So there's an incredible balance, and this is one of the big questions that virologists try and understand. And it's very difficult to study in people, so we turn to other models for doing that. And all of the twists and turns that allow viruses to propagate, whether it be in a cell or a tissue or in an entire organism or in the whole population, this provides insight into mechanisms that we couldn't study any other way. And that's another theme in this course. We can study aspects of life that you can't do without virus infections. So when this course is over, and not only it's going to be spring, which is really nice, you will have significantly better insight into how cells work and how they interact, not just viruses. Because viruses are wedges or hooks. They target processes to their advantage. So when I explain how a virus replicates, you're going to better understand the cellular process. You're going to have a better knowledge of the way information is stored and decoded in viral genomes. And this is far more diverse and complicated than occurs in our cells, for example. Many, many different strategies. We have very tiny genomes. How can you pack so much information into a tiny viral genome and have it work? We also will have a basic understanding of viral pathogenesis, which is the process by which viruses cause disease and infectious disease in general. You'll be able to better understand what's happening to you when you get sick from a viral infection. And when your family says to you, should I be vaccinated, you'll be able to make a better decision about that as well. So why do you feel badly when you get a viral infection? Immunology will explain that. And that's one of the things you'll be able to do when the course is over. In addition, you'll be able to defend these assertions. Well, you may disagree with me, which is fine, but I think you'll see by the end of the course that viruses have more biological diversity than all the rest of life. Well, not life, because I don't think they're living, but let's just say the bacterial, plant, and animal kingdoms put together. Far greater biological diversity. We are actually not at the top of the food chain. I think viruses are. I think the tiny may well inherit the earth, although they can't get rid of us, otherwise they wouldn't be able to propagate. And despite being very small, most viruses, it turns out, have less than 15 genes. We have tens of thousands of genes in us. Some viruses only have one gene, and that's enough for them to propagate and be very successful. They excel at survival in what is a pretty harsh world, and the molecular biology is very sophisticated. And finally, virus-host interactions define interactions of fundamental and practical significance that we really need to understand if we want to protect against serious virus infections. We have very, very rudimentary knowledge of how viruses and hosts interact, but we're getting better at it, and I hope to tell you some of the results that we've had. Finally, I want you to come out of this course and be able to look at a news report like this terrible one from CNN, which focuses on all the sensationalism of swine flu. You'll be able to read newspaper articles, online articles, and blogs and websites, and you'll say, this is nonsense. This can't be, it's just sensationalism. And remember that newspapers and TV programs have to be sensational to make money. For some reason, that's what people respond to. So here we have a CNN headline from actually one of my readers on the blog sent this to me last year. This was from last summer, I believe. A paper came out in the New England Journal of Medicine where they had infected ferrets with the new pandemic influenza virus. And so they put this headline up without telling the listeners that this referred to experiments in a ferret. And it can ravage the lungs, spreads through respiratory systems, causes lesions, doesn't stay in the head like seasonal flu. I don't know what that means because flu typically is a lower respiratory tract infection. The last point, survivors of 1918 flu are immune. That's true because the virus is quite related antigenically to that, and we'll talk about that. But an animal model is great for understanding aspects of virology, but it turns out that the virus was quite benign in people for the most part. And so the animal results did not predict what was happening in people. But it was just a splashy, sensationalist headline. And this is what we're trying to combat. So I want you to be able to look beyond this, look through this kind of sensationalism. So we're going to talk mainly about animal viruses in this course. but we will talk about some viruses that infect bacteria because they are great model systems. Not a lot, but enough to explain certain principles. And many viruses we'll not pay any attention to, sadly, because of time constraints. But once you take this course, you'll be in love with virology and you'll be following viruses for the rest of your lives. So I'm not worried about that. So let's talk a little bit about viral origins and history. How old are viruses? Well, with a few exceptions, and we'll talk about the exceptions, there's no fossil record. So it's very difficult to know how old they are. You can get estimates for how old they are by looking at the sequence change. So viruses change in their sequence a certain amount each year. It's called the molecular clock. And you can back calculate using a molecular clock calculation. And for some viruses, it places them among the dinosaurs, 150 million years ago. And this is all possible now because we can sequence viral genomes, and there's a whole new area of bioinformatics that uses mathematical algorithms to do these calculations. In theory, as I mentioned earlier, viruses could predate cellular life. They could have existed in a soup. We'll talk a little bit about those early origins, but my feeling is that probably viruses were around even before cellular life evolved, but I think it will be hard to prove. We do have lots of early records of virus infections in prehistory. All right, so let me tell you a few of those. So there are ancient references to viral diseases. So here we have from 700 BC from the Iliad. And it says, here this firebrand rabid Hector leads the charge. So rabid, referring to this virus disease passed on to humans by rabid animals. And they didn't know it was a virus at the time, but it meant that viruses were around. And here's another one from about 1500 to 1300 BC. It's an Egyptian carving, which shows this young priest with this leg, it's called drop foot, and a withered calf, which is very typical of polio. So we think this is the first recorded characterization of poliomyelitis, which is also caused by a virus. So the historical records are full of references to diseases which, when we look at them now, look very much like either viral or bacterial diseases. And there are entire books written on this subject which discuss, for example, how battles were lost due to infectious diseases. Now, of course, no one knew what these were, but clearly the virus infections were around as long as people were around. You can also understand from looking at the way people prevented virus infection. So as long ago as the 11th century, there were practices in place to prevent virus infections. Again, it wasn't known that they were virus infections without knowledge of the agent. And a very famous practice was called variolation. And smallpox is a disease that we can very clearly see in historical records because it has very prominent symptoms. It has a very prominent rash, it's fatal, and so we can track it historically. What was noticed very early on was that once you got smallpox, if you weren't killed by the disease, you survived and you never got it again. So people were smart. They said, I wonder if I naturally infect myself or give myself this disease, can I protect myself? So the practice of variolation arose where they would take the pustules from a smallpox victim, and these are full of virus. They would grind them up and inject them into someone else. And if you didn't die from that, because you did often, you were protected against the disease. And this is actually something brought to the UK, England, in the 1700s by Lady Montagu, who was the wife of the Turkish ambassador. And she went to Turkey and found this practice being done, where you would go to a woman who had a little room, and she would have a vial full of ground up crusts from smallpox victims. And you would give her a vein, and she would shove some into it. And as I said, that has a lot of virus in it, and if you didn't die, you would be protected. So these are the first examples of immunization. But it wasn't until the later 1700s that Edward Jenner established true vaccination. Jenner realized you can't infect people with the agent that causes the disease. He didn't know what the agent was, but he knew there were forms of the disease that were milder, so his idea was to use that. And when we talk about vaccines, we'll go through that. Now, the concept of microorganisms is not developed yet at this point in history. And really the first beginnings were from the Dutch lens maker, Leeuwenhoek. He made lenses for glasses and all sorts of telescopes, and he made a microscope one day. You've probably seen images of this very crude microscope, and he started looking at things under it. And he found these wee animalcules, things moving around, microbes, bacteria, yeasts, fungi. And from that work, initially, became the acceptance of the idea that there are things smaller than us, microorganisms. And it took Pasteur to show in the 1800s that these are generated by reproduction. They don't spontaneously reproduce. There was a whole train of non-scientific thought where people thought that these wee animalcules, these microbes, arose by spontaneous generation. Huge debates between Pasteur and his opponents. But he finally prevailed and showed that these reproduced, not by spontaneous generation. But the germ theory of disease, which links a microbe to a disease in humans, wasn't formally established until Robert Koch did so in Germany in 1879. He said, we have microorganisms and some of them can cause disease, and he proved it. But he didn't know anything about viruses yet. These are all bacteria that we know now. Now, this is how Pasteur got around the problem of spontaneous generation. So he took a flask with broth in it in which microorganisms could grow, and he boiled it to sterilize it. And nothing would grow in it unless he put air into the neck. So any contaminating microorganisms from the environment would get in here and they would be stuck down here. They would never make it up here. And these flasks actually still exist at the Pasteur Museum in Paris. They're still not contaminated. So we did a podcast about this. There's a professor at Columbia who's a historian of science, and he talked about this in our podcast number 35. So that was the proof that microorganisms reproduced. Viruses came around about the end of the 1800s. The first virus to be discovered was one that infects plants, and this will be a theme recurring. You'll see many discoveries in virology occurred using plant viruses. This is a tobacco leaf, and it's infected with a virus that causes these mottled spots on it. Eventually this leaf will wither up and die, and if you want to grow tobacco, this is very bad for cigars and cigarettes and so forth. So the scientists at the end of the 1800s was trying to figure out what caused this. And back in those days, the standard method which Pasteur had developed would be to take what you suspect to be a microorganism and filter out the liquid, take the concentrated microorganism, and in this case, rub it on the leaf. Well, this fellow did this over and over, and what he found was that what caused the infection of the leaf was actually going through the filter. So this is what these filters look like. These are called porcelain filters. You basically had a vacuum flask, and then you have this this chamber up here. You could pour in liquid up here. That's what the yellow material is. And this is the porcelain filter, and have very fine holes in it which would stop bacteria from going through. So they learned this empirically by making porcelain with different sized pores. And so the bacteria would remain here, and what went through was sterile. And what was found with tobacco mosaic disease was that the agent of the disease was down here. It wasn't hung up by the filter. And this was a fellow named Ivanovsky who did this in 1892. He found that the agent of the disease, tobacco mosaic disease, passes through the filter. Okay, so you filter it, you take this liquid, you put it on a leaf, the leaf gets infected. And that didn't make sense to anyone because up to that point everyone understood that microorganisms were retained by these filters. So Ivanovsky said, well, it's a really small microbe, and he let it go. We cite that as the beginning of virology, but he really didn't understand that he was working with viruses. It was about 6 years later, Bayerink made the same finding. The filtrate is what causes infection of the leaves, but he said, this is a distinct pathogen. This is just not just the microbe that we've been studying so far. This is something new. He didn't know what it was, but he made the conceptual leap that this is a new kind of agent, and that's really the beginning of virology in 1898. And it was followed rapidly by a discovery of many, many viruses. And the first animal virus in 1898, the agent of foot and mouth disease, and very serious infection of cloven-hooved animals like cows and sheep and goats, agriculturally very important. And by now the key concept emerged that not only were these small agents, but if you took a filtrate and put it in new broth, it wouldn't grow. The agent would not grow. You had to have cells present for the agent to grow. Right? So the agent replicates only in a host, not in the broth that were used in those days. And it became the standard over many, many years that the filters used to separate viruses from bacteria were 0.2 micron filters. And we still use these today to sterilize virus preparations. The viruses flow through the 0.2 micron filter and then bacteria stay behind. A micron or a micrometer is a millionth of a meter. And keep that in mind, 0.2 micron, because as it turns out, now we know of viruses that are bigger than 0.2 micron. And here's a list of virus discovery, just to give you an idea of the pace of discovery, the kinds of agents that were discovered. Yellow fever in 1901, rabies, smallpox virus. There were tumor viruses discovered at the beginning of the century, chicken leukemia virus, Rous sarcoma virus, but they were done in animals, and no one believed that these had any relevance to human tumors. In fact, Peyton Rous did his work at the Rockefeller, and you can see one of these were chicken tumors, solid tumors in chickens. He would get farmers from New Jersey bringing their chickens in with big tumors, and he would grind up the tumor, make a filtrate through a small filter, and show that you can inject the filtrate into new chickens and get tumors. If you go to Rockefeller, they have a museum there. You can still see his original tumors, which are preserved in formalin. Viruses of bacteria, 1915, and influenza virus, 1933. And of course, what does virus mean? It means toxin or poison. It's from the Latin word. Now, we will often use a word in this course, virion, in addition to virus, and that specifically means the infectious particle, not the non-infectious particle. When I say virion, I mean the infectious particle. So we'll use virus a lot to mean all the viral particles, and virions to mean just the infectious one. Now, in the 1940s, the electron microscope was developed, and up until then, no one had ever seen a virus. All they could see were the effects of it on cells or in animals. And we began to get beautiful pictures of viruses of different sorts, and we'll talk about this a great deal in the structure section. But here, for example, is a bacteriophage taken with an electron microscope. Again, instead of using light to illuminate the subject, we use electrons, so it has very high magnification power. And you can see the head where the nucleic acid is, the neck and the tails. I showed you that earlier. This is tobacco mosaic virus, the first virus discovered. And it's a long rod-like virus with nucleic acid wrapped up in it. These are rabies viruses, bullet-shaped viruses. And these are rotaviruses. These are viruses that cause gastroenteritis. If you see reports of a cruise ship that has to come back to port because everyone got sick and they have to get off and sterilize the ship, this is the virus that causes it. Nowadays, we know so much about viruses. We know the structures of viruses at atomic resolution. We know where every molecule, where every atom is located in 3 dimensions, and we can build these beautiful pictures of viruses on our computer. So we know the coordinate, the x, y, and z coordinate of every atom, so we can build these wonderful models, but they're static models. They only represent a static image of viruses, and they can tell you a lot, but as you'll see, virus infections and viruses themselves are not static. We even know the chemical formula for viruses. Here's the chemical formula for poliovirus, the number of carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur atoms in a virion. But as you'll see, this is an average, and you'll understand why it's an average. There is no precise formula for any virus particle. So what is a virus? It's a small infectious agent. It's an obligate intracellular agent. It has to multiply in a cell. And a parasite, as we said earlier, is an organism that benefits at the expense of the host, which is usually a different organism. My view is that viruses are not living, and you can develop your own view at the end of this course or throughout the course, because there really isn't an answer. Yes?
When you say virus particles versus virus, is a virus particle a specific part of the virus? Or you just
Well, the virus particles are these here, these images that we see through the electron microscope. That's the virus particle or the virion if it's infectious. So I think viruses are not living. They're chemicals. They can't do anything by themselves. They can't reproduce. They need a host to reproduce. So in my view, that's not living. But many people have different views, and it's hotly debated. Some time ago, I put a poll up on my blog, Are Viruses Alive? And yesterday I had 435 responses. So the possible answers were yes, no, something in between, and I don't know. And you can see it's pretty evenly split among the three possibilities. So some people believe they're living, some don't, and something in between. I feel they're non-living, but this is a great topic for debate, and I'm sure you'll be embroiled in it as well. So you should go to this web page and take this poll. I'd be curious to see if it shifts. Now, viruses are generally very small. Here is a carbon atom here, and to scale a ribosome and a poliovirus particle. So polio is about the size of a ribosome. Here's a bacteriophage, which is considerably bigger than polio. So now the carbon atom here is in this panel here. So this whole part has been shrunk so that the sizes are correct. Here's the carbon atom here. Here's poliovirus, a bacteriophage, a bacterium, which is much larger, tobacco mosaic virus, and HIV-1. So you can see they're quite small. Here's another view showing virions next to a cell with its nucleus and intracellular structures. These are herpes virions, the larger ones magnified here, and poliovirus. So polio is about 30 nanometers in diameter, slightly larger than ribosomes, and herpes about 10 times bigger. So they're very small. How many viruses can fit on the head of a pin? Of course, it depends on the virus, but rhinoviruses, which are about the size of polio, about 500 million of them. And when you sneeze, you put out enough rhinoviruses to infect thousands of people. And we'll look at that later on as well. Now, the whole idea of viruses being very small has been challenged by the discovery of mimivirus, the largest known virus, which lives in amoebae at the bottom of water, standing water. The particle is 500 nanometers, and it has fibers emanating, 125 nanometer fibers coming out, for a total size of 750 nanometers, or 0.75 microns, considerably bigger than the 0.2 micron filter that we use typically to separate viruses from other forms, other microbes. So we missed the mimiviruses for many years because they were retained with the bacteria, and now we know where to look. The genome of this virus is the biggest known, 1.2 million bases. It encodes 1,260 proteins. And these are a list of proteins involved in various cellular processes: translation, DNA synthesis, DNA repair, polysaccharide synthesis, not present in any other virus genome. We have no idea why they're there, but we'll talk about some of the possibilities. And these viruses really challenge the frontier, the border between viruses and cells. And this is what a mimivirus looks like. You can actually see these particles by the light microscope. This is an EM of an infected cell, and these are mimiviruses maturing in the cell. But you could see these in a light microscope. They're that big. And there's a website called giantvirus.org, so you could explore all this. Yes?
In this image, is the host cell living? Or is that like the virus starting to put itself out there? Because it doesn't have a smooth cell membrane.
It could be. I don't know what the uninfected cells look like here. So they might look similarly, or they could be blebbing. They could be undergoing apoptosis. These viruses mature intracellularly and then go to the surface and bud out. So I don't know what the uninfected cell looked like in that case. So viruses have DNA or RNA in their genome. In the cell, the appropriate cell, the genome directs the synthesis of everything you need to make new virus particles. And these are formed by de novo assembly of new components. You make the parts and you put them together, and then the progeny transmits the genome to the next cell or the next organism, and the whole cycle begins again. The particle comes apart, the genome goes into the cell, and you initiate an infectious cycle. We will talk about this in great detail. And remember, an infection of a virus infection of a cell is an exercise in cell biology. The cell is absolutely essential. All of these cellular functions are needed for viral propagation. Translation of the mRNA, energy, viruses can't make energy, enzymes for replication and assembly, the transport pathways. Viruses need to get in through the plasma membrane. They need to get to the right place in the cell. Some of them go to the nucleus. And then once they're assembled, they need to get out again. They take advantage of cell transport mechanisms. And we've learned so much about how the cell does these things from looking at virus-infected cells. We'll talk about virus assembly quite extensively. Viruses replicate by the assembly of preformed components. This was a puzzle to many scientists who studied viruses in the early 1900s. They were used to bacteria, which divide by binary fission. Viruses don't do that. You take a single bacterium, you put it in a broth, it splits in two, those each double again, and so forth. That's binary fission. Viruses don't do that. They make the components and then assemble the final product. And we'll talk about how that's reflected in these growth curves in a couple of days. Viruses are classified very much like other organisms. You know the classical Linnaean hierarchical system, which is shown here. Viruses are put in orders, families, genera, and species, and they are put in these divisions according to a number of characteristics: the type of nucleic acid they have, the symmetry of the shell, whether or not there's a lipid membrane around the nucleic acid, and how big the virion is. Nowadays, though, we simply sequence viruses and place them in families. So genomics has really superseded all of this classification. We sequence virus and we say, aha, it's an influenza virus, or it's something entirely new. And here's an example of that. This is an article from MSNBC, which reported the finding last year that a lake in Antarctica is full of viruses. And what they did was they went down to this freshwater lake, drilled a hole in the ice, and sampled the water, and they just sequenced it. They filtered it to get rid of bacteria, and they sequenced it. And they found tons of viruses, some of them not previously identified. So they found nearly 10,000 species of viruses, including some small DNA virus that had never before been identified. Viruses from 12 different families, some of which may be completely new to science, which means the sequence doesn't look like anything we know. So I wonder how many places are like this, how many new viruses we have yet to find. And again, this is all thanks to genome sequencing, because we can't grow these viruses, but we can sequence the genomes in samples. Here's an example of a classification of viruses in a specific family. Virus families are ending with viridae, picornavirus family, or picornaviridae. They're classified according to their genome, which we will see shortly. This happens to be the picornavirus family and the genus Enterovirus, and within that genus there are a variety of different types or species of viruses that cause different diseases. And again, these are classified by the comparisons of the particles as well as the genomes. Now, the classification of viruses is carried out by the International Committee on the Taxonomy of Viruses, and they maintain this website, the virus database, which is actually maintained up at Columbia. And you can go here and find any virus that you'd like, information about its structure, nucleic acid sequence, and links to articles on it. So far, we have identified 40,000 virus isolates from all forms of life, 1,950 species, but this is just the tip of the iceberg. We have millions of viruses per mill of seawater, the lakes in Antarctica, and who knows where else there are viruses unknown. So this number is minuscule compared to reality. Now, I want to end up by talking about the genome of viruses. In the 1950s, the viral genome was shown to carry the information needed to make new viruses. Now, this may seem obvious to us now, but in the 50s, some people thought that the protein coats of viruses were actually the genetic information. And here was a classic experiment done in the 1950s using very simple viruses of bacteria. The question is it the protein or the nucleic acid that's making new viruses? So Hershey and Chase in 1952, they prepared bacterial viruses, bacteriophages, which were radioactively labeled either in the protein or in the nucleic acid, in the DNA. They let these viruses attach to bacteria so that the nucleic acid could get in, and then they sheared off the phages by simply mixing this in a Waring blender. And then they asked, where does the radioactivity go? Well, if they used phages labeled in the protein, the radioactivity was never associated with the cell. But if they used phages labeled in the DNA, they saw that the DNA went into the cell. So this was the first proof that the viral genome encodes genetic information. Now remember, all viral genomes must make RNA that can be read by host ribosomes. Everybody follows this rule, and this will make your life much easier, because even though there are thousands of different virions, there are only 7 different genome types, and we can use that to classify viruses and understand how they replicate. So these are the 7 different genome types, which were assembled by David Baltimore, the Nobel laureate. He actually missed one when he discovered this. There was a virus family not known at the time, which were the hepatitis B viruses, which have an unusual gapped DNA structure. But all genomes fit into one of these categories, and they all lead to mRNA. So you can use this diagram called the Baltimore scheme to figure out any virus, how it's going to replicate in the cell. And we're going to use this next week to discuss how each of these genome configurations multiply. And when we do so, these are the definitions we will use. We'll often talk about plus strand and minus strand viruses, and plus strand is just the mRNA strand, the same polarity or the same strand as the mRNA can be translated. Minus strand is the opposite strand. We're going to use that a lot. So knowing just the nature of the genome, you can predict all of the steps that the virus has to undergo in order to produce mRNA. So that will really help you a lot in understanding how viruses multiply. And so these are the 7 classes of viral genomes: double-stranded DNA, gapped double-stranded DNA, single-stranded DNA, double-stranded RNA, single-stranded RNA of both plus and minus polarity, and finally single-stranded plus RNA that goes through a DNA intermediate. The 7 classes of RNA. Viruses can have any of these, and of course we just have pretty much double-stranded DNA, so we're rather simplistic. So next time we'll start to go over how each of these move into mRNA to help us understand replicative cycles. All right, thank you.